Welcome, everyone, to today's special episode of the Destroy Duchenne podcast. This episode is a little different from our usual ones because just a couple of weeks ago, I joined Somite Therapeutics as a consultant. I'm really excited about the work they're doing, but to be honest, I still have so much to learn about this incredible company and their groundbreaking approach to medicine. So today, I'm on a journey to discover Somite, running alongside all of you. What drew me to Somite was their AI platform, AlphaStem, which has the potential to revolutionize drug development and cell therapy. As a huge advocate of AI in the medical field, I believe it's critical to harness the power of AI to advance treatments, discover new therapies, and ultimately improve and even extend human life. Ever since I was a kid, I've been fascinated by technology. I looked up to Steve Jobs and would eagerly watch Apple releases, even if I had to do it from the back of a classroom. My favorite business role model is Jeff Bezos, who I had the pleasure of meeting. Like Bezos, I believe in the transformative power of technology. That's why Somite's mission and their use of advanced AI resonates so deeply with me, and I can't wait to share this journey of discovery with you all. Now let me introduce you to the remarkable team at Somite who are pioneering this incredible work. Micha Breakstone, who is the CEO and co-founder. Micha is a serial entrepreneur recognized for creating groundbreaking AI-driven companies in healthcare and technology sectors. He's the co-founder of Chorus.ai, an AI-powered conversational intelligence platform that was acquired by ZooInfo for $575 million. And he founded NeuraLight.ai, which he raised $30 million in a year to advance AI solutions in neurological health. He holds a PhD in cognitive science and a master's in mathematics, making him a visionary in applying AI to solve complex healthcare challenges. And then we have Dr. Jonny Rosenfeld, who's a co-founder and CTO. Jonny is an AI pioneer and strategic leader in a large-scale machine learning and AI research with a deep impact across fields like genomics and information theory. He leads the fundamental AI group at MIT FutureTech, bringing over 20 years of experience in AI, engineering, and interdisciplinary science. Known for industry-defining breakthroughs in AI, he has received prestigious awards, including 2 Israeli Defense Prizes, which is like the USA's Medal of Freedom for 2 projects he led. He holds 5 degrees, including a PhD in AI from MIT, and has directed innovative research that continues to push the boundaries of AI applications. And then we have Dr. Kristy Brown, who's a Senior Vice President of Translational Development. Kristy is a leading expert in translational drug development, known for her success in advancing therapies from early research stages to clinical trials. She's the former VP roles at Rejuvenate Bio and Biosciences, where she specialized in preclinical and translational science for breakthrough therapies. She holds a PhD in analytical chemistry and brings extensive scientific rigor and strategic oversight to Somite's DMD program. So these people are obviously very, very accomplished. Very grateful to have you all here. And really, my first question for you all is, how in the world did you guys all get connected? Because you guys are so highly talented and skilled. I just got to know how this all came to be.
That's easy. That's Micha.
So first, just listening to you kind of read Jonny's and Kristy's background, I was like, wow, I get to work with these people. So it's always humbling. And Elijah, thank you so much for having us. In terms of founding story and how we kind of met everyone, so it's a long story, but the kind of bullet points are, I was looking to found my next company, and I reached out to a dear friend of mine for the past 25 years, who's a professor at Harvard Medical School, and I asked him if he had anything interesting at the intersection of AI and biology therapeutics, and his answer was, Micha, let's stay friends. So naturally, as an entrepreneur, I had to ask another 3 times until he finally kind of acquiesced in introducing me to Cliff Tabin, who's the chair of the genetics department at Harvard, and Olivier Perquier, who's another National Academy of Science member and faculty at Harvard Medical School, and later on also Jay Shindure at University of Washington, who's one of the pioneers at single-cell RNA sequencing at high throughput. In parallel, 2 things happened. One is I was introduced to Jonny by several of Israel's top people, including the entrepreneur of another incredible company called Immune AI, so a kind of a billion-plus AI for immunotherapy company reached out to me and said, hey, you got to meet Jonny. Naturally, I didn't see the email, so I had to wait for another 2 people or so, or at least one other person to nudge me. And finally, I took the call with Jonny, and I'll let Jonny tell his side of the story. From my side of the story is like, oh wow, kind of what an incredible guy. And I'm used to be able to master kind of AI in the conversation. I was like, oh, I have to learn from him. So that was on the one hand. And on the other hand, I was introduced to Ilan, who is the CEO of Somite, and he introduced me to the person who's currently our chief science officer, Carl, and we were talking about kind of the prospects of changing or advancing and making progress for Duchenne using this new modality of cell therapy. And I was like, okay, so who's the guy that can actually make this work? Little did I know it was not a guy, but a girl. And and immediately they both said Kristy, and they lit up. So I said, okay, I have to meet Kristy, and kind of that was, I believe, December of last year, December of 2023, if I remember correctly. And then just getting these incredible people in the room together, there was very, very little to do than let them speak with each other. So that's kind of my point of view. But now we can hear the what other people think the way we met was.
Well, I can chime in on my side. Micha is one of the top entrepreneurs in Israel. His name reached far and wide. I was looking, to make it short, to found a company. I think it was the fifth or fourth or sixth time that I heard the name Micha Breakstone. And the second or third intro when he finally took the call. And as the saying goes, the rest is history.
Kristy, how did it look from your side? Sorry, Elijah, I'm taking over for you and asking questions.
No, please.
No, it was great. So most of my career, I've been studying muscle and Duchenne specifically, and I had recently left that space, and I wanted to come back. And so being introduced to a company and a CEO who wanted to drive the therapeutic forward, it was where I wanted to be, and it was a good opportunity and good science, and I get to learn on the AI side as well. So it's always a great career opportunity when you get to drive programs and continue to learn.
So, like Kristy, I was speaking to Solid Bioscience, and that's where I met Kristy, and I worked as a consultant along as Micha said earlier. That's who brought me into the biotech field. So I have a love for that company and those people there, and so that's all pretty cool. But yeah, so let's get into it. I have some hard questions, if you don't mind, but I want you to react to this. So Noam Chomsky, if I say his name right, he's a renowned philosopher. Chomsky, yes, Noam Chomsky. Thank you. Is a renowned philosopher in conscious science, and he said this about AI, and I want to quote it and get your reaction. So I quote: "The human mind is not like ChatGPT and its ilk, a lumbering statistical engine for pattern matching, gorging on hundreds of terabytes of data and expectulating the most likely controversial response or most probable answer to a scientific question. On the contrary, the human mind is a surprising, efficient, even elegant system that operates with small amounts of information. It seeks not to infer brute correlations among data points, but to create explanations. Let's stop calling it artificial intelligence then, and call it for what it is, and make plagiarism software, because it doesn't create anything but copies existing works of existing artists, modifying them enough to escape copyright laws." So given this critique, how would you define AI, and do you agree or disagree with his assessment of AI as merely a predictability machine?
I'll let Jonny answer this first, but before I let him answer, I'll just say that the question is so good. You didn't prepare us. Usually people are not as intelligent as you are, and this is a wonderful, wonderful question. It also touches very close to home. So Chomsky was the mentor of my PhD mentor, actually. So I'm his academic grandson, if you will. And Chomsky's opinions themselves have been have shifted over the last 30 or 40 years. Not very much, I have to say. He's also kind of also one of the most quoted in history, cited in history, academics and researchers, but also one of the most profound ones. So I have a lot to say, but only after Jonny addresses the AI part. I'll address the cognitive part.
Okay, so I'll say so Chomsky is a living legend, which doesn't take away any one bit, quite the contrary, from the acerbic tone or the controversiality of the opinions that he holds. So without talking about the persona too much, I think it's worth dwelling on the critique and what we and this hazy notion of AI, sort of sort what are these systems that we are talking about. So just to explain to the broader audience what the critique actually means is the likes of ChatGPT and the models that we encounter today are trained on vast amounts of data. So this could be, the entirety of Twitter, formerly known Twitter, currently X, or Wikipedia, or the body of digitized books, and articles and transcribed YouTube videos, and basically whatever you can put your hands on publicly and otherwise. And the critique goes, just to put it in layman terms, well, these things, one should not confuse reasoning and intelligence with the ability to regurgitate things that they were already trained on. So that is the essence of that critique, at least as I hear it. And with that, the counter would be to question, okay, how can you tell apart this idea of I'm just regurgitating things that I already heard, and I have the entire internet and the X-verse at my fingertips, and I already memorized it, versus I can reason, I being a model. And this has been called, so Chomsky is not special in this critique. This has been called the stochastic parrot sort of paradox. These models just spewing the most likely next token or word. The counter, so if we have this notion of just regurgitating knowledge versus reasoning, we can actually test that very well. You can construct an example where it is guaranteed that the ability to generate the next token or word or sentence or action would not have occurred or is almost guaranteed not to occur in the dataset. And indeed, when you test these machines, these models, in their ability to create novel outputs that were not seen in the dataset, you can find them. That is what is known as out-of-distribution predictions. It doesn't always, the reality is more nuanced than that. There are things where these models excel out of distribution, and where they fall very short. They can, if you will, only interpolate or cross in between the dots that they already saw. So both things actually are happening at the same time. We're moving towards more capable reasoning models, which are not just trained, but actually are now at inference as they generate their answers, need to examine their answer and actually reason on top of it in order to generate and fix their answer. And there are even, I'd go as far as say today, early agentful systems, the likes of early software generating systems or protein design systems, if you will, that will be relevant. And not too long ago, a Nobel Prize was won on, that are designed such that they iterate on the reply. So to Chomsky, I would say, you are stuck in the stochastic parrot argument, but you have not considered, or maybe we should talk about, the Kolmogorov parrot, which is one that can create algorithms that generate the patterns that we're interested in.
So you're basically saying that basically some of the newer things that are maybe not so popular or seen, that's not mainstream yet, is that you can actually have models that has more resemblance of true intelligence, where it's not just going off of the data sets, but it's actually able to come up with novel ideas, essentially. Is that correct?
Yes, I'll refine it slightly because intelligence is a slippery term. So deep learning or the current AI is not a new name, and we have progressively been moving the goalposts. So in its current form, the latest sort of advancements in AI are just yet another chapter in a long scientific field, long existing, of what you might call the science of learning. Okay, and now intelligence, I would suggest instead of asking what intelligence, it's almost philosophical. Like, let's consider tasks. So we can consider the breadth of possible tasks that humans can, in principle, tackle, and then tasks that humans cannot tackle. Like we are not especially good in designing proteins from sequences, and yet we have built successfully models that, as I alluded to, and I expect we might discuss, won the Nobel Prize for building blocks of life. So both tasks that we are well versed in as humans and tasks which we are not, you could ask, how well can a system that can be taught or can learn, how performant is it on any given task? And that becomes something that we can very well measure. And we've been making a steady progress on it. And we can even go as far as say, how does the amount of data or the amount of computation that goes into these models affect the level of performance that we can expect? By now, we know how to answer these questions very, very well. Part of that is the research that I have done at MIT.
So, Elijah, I have a lot to add, but I don't want to take over the podcast. So tell me, should I answer in 60 seconds, 120 seconds, 120 minutes? You tell me.
For the sake of time, I guess let's really drill on this point because I think it's a big one. But then the other questions after this, we can maybe I'll give you a timer for that because we don't have unlimited time. I wish we did, but let's really go into this one. So far away because I think this really sets the foundation.
Okay.
For what we're talking about, because something I want to make clear to the audience is, when we saw OpenAI, rise up, I don't know, 2-3 years ago, every CEO in America claimed they're an AI company. And so I want to show that, this is a true AI company. This is not just slapping the label on to look good for investors. So go for it. Yeah.
Yeah, that is for sure. But and what we're doing here is kind of a philosophical or meta level. So I will shorten it a bit because I could go on and on and on for a long time. So first, I'll say that when I introduced Jonny, I failed to mention that he introduced I guess two really seminal concepts in AI. He was very bad at trademarking, so he doesn't get a lot of credit for it. But the first was he was the first to model scaling laws, or in simple words, how much data you need in order to achieve X improvement on a model. Basically turning this from a black magic to an engineering problem, which is a claim to fame that he had. He published, I think, in 2017 or 2018 when it was still really black magic and nobody knew this. He just didn't use the word.
The first paper on scaling laws from our, this was not only me, but also others at MIT. It was in 2019. Then the world exploded later in 2020 with OpenAI to a very large degree, showing this at scale and very likely concurrently doing this as scientific breakthroughs happen and discoveries. But this was, so this really...
And he'll say science is always a community, which is true, but he also was a key figure there. And the second is the notion of what Jonny has come to call the scaffolding hypothesis, which is hyper related to kind of agentive AI or iterative AI, as he posed. And so with setting that stage, it's a little hard for me to say, well, I completely disagree with Jonny, but I do. So in a sense that I think that Jonny is actually right in everything that he said, but I think there is also a philosophical kind of component here, and I'm much more on Chomsky's side. So I'm much more on the fact that you won't be able to actually create novel intelligence or novel concepts by regurgitating data. That's just my own opinion. I don't think, I think it could be a scientific question, but currently it's posed as a philosophical one. I do completely agree with everything that Jonny said in terms of the analysis of the situation, of course. And I think, kind of TBD, one thing we do know, and I'm sure Jonny will agree with me, is that everybody agrees that training, we're hitting a limit on what we can train on, right? So there will be at some point an implosion, right? You can't train on data that is generated by AI. You'll get zero upside, right? So, and we're hitting our limits there. So at some point we'll need to have novel ways of generating data, by the way, not only in natural language, but also in cellular data, which I hope we'll get to later on. So kind of looking forward into the future of this conversation, kind of 20 minutes out, when you ask us, like, is this real AI? The answer is hell yes. It's not only real AI, but we own the AI because we're able, together with two incredible people that happen to be sitting on the podcast next to me, both Kristy and Jonny, being able to actually generate the data in a format that I don't want to steal their thunder, so I'll let them speak about it. So when we think about this, I'm much more on the Chomsky side, namely intelligence is magic. It's not enough to spit in an infinite amount of text and hope that insights come out of there. It's kind of a philosophical belief, like great mathematicians. I don't think it's just a matter of just throwing in a lot of data, but this is really a philosophical question. I know Jonny does believe, right, that consciousness and intelligence is just a matter of compute and data, right? Jonny, I'm oversimplifying, but I'm guessing that it's
yeah. I feel an urge to add some nuance. So
yeah.
I don't think anybody is on, well, the camp of if you throw enough data at it and don't so do like so nobody is claiming that the path to intelligence is regurgitation. Scaffolding hypothesis, for example. So we already have superhuman for a while now, chess players and Go players, right? And that came about through self-play and actually algorithmic improvement through iteration, right? So it's very clear that being superhuman at a given task is possible. Like it's like there we did it, and we keep doing it. Now you ask for any task that a human can do, and there again, models on their own are not the solution, but systems that have models. Now we can argue. I don't think that there's anything magical about intelligence. We are computational machines. No more than there's something magical about a living cell, which is also a computational, extremely concurrent, running multiple programs in parallel computational system. It happens to be systems that we don't understand very well yet.
So I think for the sake of time, I'll move on. But I have to say, we're gonna have to do another podcast one day because I would love to go deeper into, like, I'm a big philosophy guy, so like Jonny and Micha, are like your thoughts on free will? Because I just kind of heard, Jonny, what you were saying there, and like what's your thoughts on scientific determinism and all these, because I think that leads to a lot of this, because it is like a philosophical question. So I'm fascinated by this. But yeah, I mean, I should have asked for 3 hours. I only got an hour and 15. But for the audience, go ahead, Micha.
I was just gonna humoristically saying for the audience just tuning in, to summarize what have been said up until now. The way I would frame it is, Jonny believes we don't have a soul. What?
I've been what? It's just of course we do. It's just that it's a byproduct of a computational process.
And now you'll see my role at the company. We're going to move on to the next question now.
So, for the audience listening, because I know this is the Duchenne podcast, we are going to talk about Duchenne, I promise. I do have more AI questions, but hopefully we'll go through these faster. But I think Dr. Rosenfeld was saying, I think he was alluding to this. I want to talk about AlphaFold, which is inspired, I think, AlphaStem, which is what Somite is working on. So AlphaFold, developed by DeepMind, revolutionized biology by accurately predicting protein structures. which was once a highly time-consuming process. Somite's AlphaStem platform appears to be similar breakthrough, but in cell therapy, using AI to predict and control cellular differentiation. So can you explain how AlphaStem works similar to AlphaFold, and what role does it play in making cell therapy develop faster and more precise? Whoever wants to take it.
Yeah,
Probably
I
for
can
Jonny
talk
to
about
take
the
it.
technicalities a bit and then hand it over to Micha
Let's I
and
want
Kristy.
to, yeah, Jonny, just take 60 seconds and answer. I think let's hit the right cadence there.
Okay.
That's my idea.
So AlphaFold is truly a breakthrough in the sense that what was an extremely hard task to scalably solve, which is protein folding from the sequence of amino acids, is now scales very well with compute and the amount of data. And protein structure, obviously, one can think of it as structure. So you can think of it as lock and key sort of a functionality, and the electrostatic distribution is another aspect of it. But in principle, these are key components, building blocks of life, if you will. But there's another layer on top of it, which is systems of building blocks and how they interact. One of these, and how do they execute programs? One of these programs is how do cells actually decide what type of cell they become? Right. So we all start with a fertilized embryonic stem cell. That program of then proliferating and splitting into cells, that some become neurons and some become a muscle cell and some becomes the retina and some become skin. That has without fail a program that has been running, depending on how you count, a good many millennia in humans, and then in mammalians much more. How can you build a model? Can you build a model that now mimics or controls the language by which cells communicate in order to choose what their fate is? That is in essence what AlphaStem is. So think of the equivalent of not designing the building block and not learning human spoken language, but rather learning how cells change their programmatic state mediated by their signals, their communication, the equivalent of their words.
Does anybody else want to chime in on that, or?
From my side, no. I mean, I think Jonny answered perfectly. Kristy?
I do have a follow-up question. Maybe Kristy, you can answer this, or whoever. But something that I think is highlighted that I've seen on the website is the digital twin. Can somebody explain what that is, why it's significant, and what's going on with the digital twin?
Yeah, I'll take that, and I'll also draw an analogy kind of to what we're doing, kind of what ChatGPT and more standard AI looks like. So digital twin is just a computational model that seeks to simulate the constructs of how an embryo develops, or rather subcomponents of the embryo, because it's not the full embryo. And in a way, it's basically just scraping out all to start off with, at least, all of the publicly available data out there, and putting together kind of the infrastructure of what it looks like for cells to differentiate over time, create tissues to evolve into different organs, etc. So that's basically a digital or computational model of the either human embryo or different embryo or subcomponent thereof. I think what's interesting and why this is important for AI, and I know you kind of want to focus there. So when we think about how we actually turn this into a usable computational model, namely how we learn to push a cell from state X to cell Y, like what we need to do in order to take a cell and turn it, for example, into a satellite cell that then we can thereafter use in order to try and alleviate patients with Duchenne and other dystrophies, is kind of how we need to learn more than kind of a snapshot of the world, which is so. A good analogy would be when ChatGPT was training kind of they downloaded the entirety or the entire corpus of the internet, which is fantastic because it's cheap, relatively cheap to do that. On the one hand, on the other hand, and it's massive on the one hand, and on the other hand, it's unfortunately not very good at specific tasks. So it's very hard to download the entire internet and expect the model that you're training on to be able to translate well between two different languages. In order to do that, for specific tasks, what ChatGPT did is they added also very high quality, much more expensive corpora, which were fully aligned, say, from English to French, right? Like tens of thousands of sentences that were perfectly translated by humans between these two languages, and every set thereof of two languages, right? Or many sets of languages like that. And that kind of data is much, much more expensive. So when we think of the digital twin of the embryo, we have the component that is quote unquote cheap, where we're just going and harvesting all the data out there, single cell RNA sequencing and other modalities are just publicly distributed. And then we have this capacity to not only do that, but actually catch more than a snapshot of what's happening to the cells, but actually transition states. So take a state from what happens when you feed a cell at state X. Jonny was referring to this earlier as every cell is a computational model, right? So like taking a cell as a computer, feeding it with input A, what happens to this computer X? Like how does it turn into X prime? One of the things that we're driving at Somite is, so you can imagine that, so going back to the French example, when you feed in trillions of tokens that have nothing to do with English and French specifically, and that add on this highly curated, really, really high quality of tens of thousands or hundreds of thousands of sentences, suddenly you get this magic because you're working back on the scaffolding of general language capabilities. You have this highly aligned, high quality English to French, and suddenly ChatGPT is able to perfectly translate between English and French, which is like, it's basically like magic. Going back to the cellular example, you have this digital twin, which is the background model. And one of the things that Jonny and Kristy are truly pioneering at Somite, is the ability to generate the equivalent of highly accurate French to English corpora, but for cells, so transition maps for cells. It's not novel to be able to do that. It's novel to be able to do that without going bankrupt. So these things are very, 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 very expensive. These experiments, these experiments cost millions and millions and millions of dollars to be able to generate anything of significance. What Jonny and Alon and Kristy are doing are incorporating methods to allow to reduce the cost by 3 orders of magnitude. We then marry this extremely specific and insightful kind of model of transition maps, marry it with this background of the digital twin, let them play together, where the game is not win a game of Go or win a game of chess, but find the path to transition a cell from state X to cell X prime, play against itself, and you come up with this amazing model that we will have hopefully unleashed to the world in a year or so. Sorry, that was a very long-winded answer. I hope that was helpful.
So if I'm understanding correctly, basically the digital twin is basically mapping out how cells differentiate or how they communicate with each other, which could then lead to the correct differentiation that you want for the tissue that you want it to eventually become. Is that correct?
That is definitely correct, but not complete. So, but it's yes, it's definitely a true statement.
I'll say it maybe like so. So imagine, so because natural differentiation happens without fail, then mapping that in greater detail gives you the equivalent of a map, right? How do cells go through the different states when the natural program of differentiation is executed? Now the turn-by-turn sort of signals, if I want to get to a neuron and not a muscle, that signal is missing in the map. So Micha was discussing how can we turn on a signal or that turn-by-turn instruction and see where the cells go. So the digital twin is creating the map from primarily mouse embryonic data. And the complementary data that needed is the turn-by-turn instructions, which is the internal proprietary data that we are generating in a manner that now we had to make much more scalable.
And so you're getting this data though from like you said, mice and other things like that, correct? Like this is where you're getting the data.
Yeah,
Then
so note that if, at the end of the day, if we think of Duchenne and other therapies, we are interested in human cells, naturally, and human therapies. So there are two things that need to happen between the digital twin and the actual therapy. Digital twin is a map of natural differentiation in mice. The two things are we need to move to non-natural differentiation in the sense that we do it in the dish. We're literally growing cells in the lab. So that's one change. The other one is that we moved from mouse embryo into human cells. So those two transitions are where that we actually test out those signals. You literally have cells in one state, you expose them to the conditions, to those signals, and then you measure what is the resultant state of those cells.
That's this is phenomenal. I mean, I once wrote something. I posted it on my social media. But if scientific determinism is true, this kind of goes back to the philosophy. Then you could basically solve any problem you want with enough. You're talking about like super intelligence and things like that. But if we can map everything out, everything is a problem that could be solved eventually, if that's true. And I mean, what you're doing here in biology. I mean, in theory, in my opinion, it sounds like you could basically solve anything with enough data and time. Sure, it may take time to get all this mapped out, but once we have a map, we have a map, and
Yeah,
we know
so
where to go.
there's a beautiful concept and book by Stephen Wolfram, and there's one pivotal concept there that is really worth dwelling on, which is this notion of computational reducibility. Like you can't break a cipher just by trying it out. You can in principle, but it's highly inefficient. But problems that we do care about, or that evolution cares about, like how do I navigate a better and a competitive advantage throughout? So it can be thought of the more efficient search algorithm among sort of how to will win. So it's not accidental that the problems that we care about to a large degree are problems that are amenable to be solved with reasonable amounts of data and computation, even though there's a very large class of problems that do not fall into this category. So biology, not accidentally, falls into this category. AlphaFold, for example, you could ask, why would it be in principle possible from a finite set of examples of sequences and 3D structures to solve these problems, where I can prove that for any general system, it is known in physics as the many-body problem. If I just organize molecules and ask what will be the lowest energy state, that is an exponentially hard problem to solve. So the problems that we are interested in are not general. For the problem we are interested in, they have this property of being computationally reducible, and that is not accidental. Like we wouldn't have been living here if it was complete chance.
That is a lot to think about. Like I said, this podcast needs to be 3 hours, but let's pivot now. I think it's important. Let's pivot real quick. We can come back to the AI. I definitely want to come back, but I want to talk about Duchenne Muscular Dystrophy. Why we're here for this podcast, and I think Kristy. So as a consultant, I joined the scientific meetings and that she leads up, and that she works on. So I think that it makes sense to have her answer these questions. Again, everybody can chime in whenever they think. But what is Somite's approach to Duchenne? What are we doing? Let's start there. Let's get an overview of what the approach is that Somite is doing for DMD.
Yeah, I'll take that one. So in Duchenne, as you're aware, that you have chronic degeneration, regeneration of your muscles. which is done in like all muscles with satellite cells fusing and repairing the muscle. But in Duchenne, those satellite cells are lacking dystrophin, which is why the muscle also lacks dystrophin. And then it's fragile. And over time, you have an exhaustion of your satellite cells and you have a failure of repair. And so our approach is if you could make those satellite cells have dystrophin and be healthy, regular satellite cells, that when you repair muscle, you're now repairing it with healthy, fully dystrophin competent muscle. And so we want to know if we can take iPSC cells, so a pluripotent cell that could turn into any type of cell, and in a dish, turn it into a satellite cell using the same differentiation process that happens in the development of the human. So it's always goes along in its own niche, and then place that into the actual muscle. And so we want to start in the hand. We think the thinner muscle of the hand, if we replace the satellite cells there or gave them a new pool, those satellite cells can not only fuse to injured muscle and make new muscle fibers, but the pool itself will also regenerate. So satellite cells can also make more satellite cells. So it's there the next time that there's a bout of injury and repair. And so we can do this in mice. So we can grow the cells in a dish and make human muscle that is functional in a mouse. And so that would be the foundation of our non-clinical IND package. And then we want to go into the clinic. We would focus on, again, the hand, probably in older DMDs patients to retain the quality, the ability to control a wheelchair, use a device, a handheld device. And that's just proof of concept, because we're asking a lot. Can you take, a pluripotent cell, turn it into a differentiated human cell that then you put into the human and see if it makes new tissue? And so we're asking a lot of questions. And so we want to be very focused in the first clinical study. And then if we see if that works, then you imagine you can go to other muscles and scale up and scale out. So it is one muscle at a time, but it is replacing that underlying failed regeneration process.
Yeah, no,
Would
I
it be
mean, you
helpful
could get
if
dystrophin.
you want- Sorry, should I share my screen with experiment design or not?
Sure, you could try. I'm not sure if that will it show. I've never actually done that before on this Riverside, but we could try
I'll
it.
yeah. Let's see.
While you're doing that, Dr. Brown. So when it comes to delivery, though, what does that look like? I know it's early in the process, but what is the thought process around that? Because it sounds like this would be something that's done, outside the body ex vivo, but then you would, deliver it. Then, as opposed to a lot of treatments like gene therapy, where you're going in, but then it, takes place within the body.
Right. So it's not a systemic delivery. It'll be an injection directly into the muscle and multiple injections. So the satellite cells aren't something that you can put into your blood and then it finds its way to the muscle. You have to be within the muscle itself. And so it is multiple injections, not dissimilar to, say, like Botox, which does the opposite, right? And so that is a chemical injected directly into muscle. Similar size needle, likely, and multiple injections to deliver the cells. But then once the cells are there, they will maintain their own niche.
Yeah, no, I mean, it sounds like, if you could get dystrophin within satellite cells, it would be, satellite cells, they keep self-renewing and then also creating more progenitors, which then creates more muscle. So you would effectively solve both of the two issues that you noted, the degeneration problem, because now dystrophin is going to be in those new muscle fibers to stop the breakdown. But then also, too, you're going to have satellite cells that divide properly to keep repairing the muscle when it breaks, because it naturally does, even in healthy individual. So, that as a patient, that potential reality would be huge, even if it's only just certain parts of my body for now. Which again, if you could do it in certain parts, you probably could scale it up to larger muscles and to other areas as well, that are at least skeletal. So that's exciting. I wanted to ask you, how does AI, in particular, when it comes to this, to Duchenne, how does that how is that being used, leveraged to help solve this issue?
Maybe I can tell the story that. Oh, sorry. Do you want to take that?
No, you do. You go.
Sure. Like, how did this become, understanding biology of muscle into a drug, a therapy? And it goes back to how we started the podcast, which is the origin story of Somite and what minds were gathered together to launch this. So, our scientific co-founder Olivier Porquet, developmental biologist for many years, studied, how is muscle made, and he was able to redo that in a dish. A lot of trial and error. 10 to 15 years of work, groundbreaking data and discoveries, but not a drug, right? It was understanding science and biology. And when he collaborated with Alon Klein, who was our digital embryo, who we also talked to on the podcast, and they compared using single cell sequencing what he was seeing in the digital embryo to what Olivier was doing in the dish. They could see that they were driving on similar roads, but maybe Olivier was sometimes taking the dirt road rather than the paved highway. And so they were able to change some of the reagents, maybe change some of the timing, and go on those paths a lot clearer. And they doubled immediately within a few months of that alignment, which was computational biology, not pure AI at that point. And they were able to really improve this protocol. And in the process, the protocol itself became more manufacturable. We were able to replace certain reagents that are hard to source, not sourceable as GMP reagents, and replace them with chemicals that are compatible with manufacturing requirements that you need to take something to the FDA. And so now, within a few months, we took decades of research and turned it into something that's now a drug. And we've transferred this to a GMP manufacturer and producing at larger scale. And so that's how we leverage the digital embryo, brilliant scientists in the field, bringing their science together. And now Jonny sees a way that if it works on the comp bio direct approach, and we know we can improve, follow this roadmap and know which way to turn, that this can become an AI scalable problem. And so how we're using it right now in the program, I think that when you develop a drug, I mean, you've been consulting for so many drug companies, that once you lock your process and start engaging with the FDA, you can't keep fine-tuning it. And so for the first muscle program, we're almost locked, not quite. We're trying to learn as much as we can before we lock it. But we keep our eye, all the data we're making on that LAC process now circles back in and back to Jonny to leverage for our next program or the next iteration of it, new Somite lineages, and how we want to take it forward. But just because of the way the FDA works, at some point you have to pause your development and then take that to the clinic to see if you have functional efficacy.
I think that's just fantastic. I mean, that's really what I'm trying to get at too, is that, when we use technology, we can improve the quality of lives of people much faster. And it sounds like that's what you're saying.
Mm-hmm.
You're taking a lot of research and shortening the time it takes to actually, develop to overcome those steps. And I think that's how technology should be used. I think, Steve Jobs, he talked about how basically like you want to make tools for the mind or for humans, right, to use. That's how he viewed it. And I think, there's a lot of talk on AI right now, the ethics around it and everything. But I think the AI is just also a tool to aid humanity, not necessarily replace humanity, but aid them. Or perhaps even in the case of some jobs, I do think it should replace if those jobs are dangerous or, whatever it may be. But with medicine in particular, my interest, right, with what I do, and I think it leads to a really exciting future for patients like me. Going back to AI with the last, the time that we have left. What are some of the challenges with AI that we have right now? What steps do we need to overcome? Is it scalability or what would you say that is?
I can give it a first try and then let Jonny elaborate, but I think it would be impolite not to introduce our new podcast member in the audience there. So We have
That's
Biscuit.
Biscuit. He's
Yeah.
in his little chair back behind me. So sorry for the crash.
No, I think it imbues the podcast with a much with a very warm nuance there. I love dogs. I think the challenge, the biggest challenge for companies that are trying to bring in, first of all, okay, there's a positioning challenge where I think you started the podcast by saying everybody wants AI, and everybody is saying they're doing AI. And the truth is, most people, let me phrase it nicely, if the AI had three letters, they wouldn't be able to spell it. That's a nice way of saying what most people understand about AI. Going to more technical people that actually do understand it, there's a real challenge in bringing it in to companies that are doing biotech, in that the data that is needed to train real models is extremely, extremely scarce and or prohibitively expensive to generate. And I think that's the number one challenge, which is why Jonny has been banging on my desk for the last 9 months saying data generation, data generation, data generation. After about maybe 4 months of doing that, I actually understood what he was talking about and understood that he, of course, as usual, was seeing way into the future. So I think by far the real challenge is data. Afterwards, like training models, I don't want to say it's commoditized, it's not, but it's more people in the world can do that. That's kind of my take on things. But Jonny, your thoughts?
Yeah, I think it's a misleadingly deep question, in the sense that it's relevant both in our domain and very generally so. And it really starts not with AI, but the question of, is the problem that you are trying to solve well posed? And is it in principle solvable with the visibility that you have into the system that you're interested in? Like, why would it in principle be possible if you show me enough data or whatever, this type of data, to make progress on this problem? Right. So I think it has in many ways more to do with engineering or physics, if you will, whatever you want to call it, than AI. Now, AI is a tool, as you say, which happens to be a very efficient one, or we could argue how efficient. It's a very predictable one. If you have a well-posed problem that has you observe something that really determines the outcome that you're looking for. In the case of protein folding, okay, do sequences determine completely structure? Okay, fantastic. I understand. I understand what the input is. It's sequences. I understand, and there's a very finite set of amino acids. I understand what the output is. That's 3D structure. So the first question is here, okay, so what is the well-posed problem, and do we have enough observability? And to jump ahead, the answer is, well, it's the control of cells for the cell fate choice as defined by the state of RNA expression. This would be the transcriptome. And we believe the answer is yes, but not to belittle how complicated of a question that is. It's not trivial a priori that this would be the answer. Like, do the same signals operate similarly or differently on different sub parts of the differentiation tree? Okay, I could build a piece of software where there's a if statement hiding only in this branch, and you would never learn that from seeing all the rest of the branches. Okay. Biology is somewhat more compact than that. We know there's a lot of reuse. So this is a long way to say that there are many technical challenges, but the thing really that one needs to attend to is, do I understand the problem well? If the answer to that is yes, then it becomes a data and compute limited problem.
Could you use it to better understand what the problem is? Like you're talking about, like if it's not well defined, could it ever be used in that regard?
Well, so this is a bit meta. The short answer is yes. I mean, that is a task that we engage with.
We're coming full circle to
The
the
reality,
beginning of the conversation.
Right.
It's
exactly.
That's what I was thinking, because it's about like you were saying, if the data is not already there and it makes an answer that was not seen in the data set. It's kind of the same thing. This makes me wonder, and Micha, this is more of a question for you. My business mind, my entrepreneurial mind is thinking, do you ever think about licensing this technology out if it's perfected to other biotechs to use? Forget making drugs here, although that would be great as well. If another biotech, wants to use it, you license it out there, and that becomes the business. Is
yeah,
that of
100%.
interest?
No, yeah. So for sure, I'll answer in a second. I'll just also go back to the previous question and say one of the other reasons that it's very challenging to integrate AI is usually on the other side of the field, on the biotech, you don't meet incredible people like Kristy that are open-minded and are willing to kind of integrate novel approaches into their current workflow. So we've been extremely, extremely, extremely lucky to basically have Kristy and Jonny put our AI program together. So this is definitely not an AI only. Like, we could not have done this with a single discipline. We would have gotten nowhere. So basically, if we didn't have Kristy and Jonny working together on this, we would have maybe been able to answer questions, but they wouldn't be really relevant to anybody. So I think we're very, very lucky and unique as a company in that sense. To your question now, 100% yes. In fact, we were just presenting yesterday in Toronto to Omnia, which is the CDMO that is producing and differentiating our cells for us. And one of the discussion topics was, guys, look, we have this AlphaStem, currently a V0, kind of very early platform. And if we have a minute, happy to also let Jonny demo it if you'd like. And one of the things that we aspire to is actually completely give it as an open access tool, just like AlphaFold has done, right? AlphaFold has put this up on a server and let researchers use it for free. And our goal, both as a business, but not only, also as a service to humanity, is to put this up and make these tools available to every researcher. Of course, we will make money off of it once they try to commercialize it, or if they want to manage service. And as your entrepreneurial mind was very kind of wise in seeing, of course, it's a huge driver of revenue and more than revenue, partnerships, right? Which is 100% what we want to achieve. But we're also extremely happy to give this out for entirely free for researchers in academic setting, and helping move the entire field forward. So 100% yes. And the best example is AlphaFold and Rosetta Fold that have done incredible work in that regard.
I think that's absolutely beautiful, like business-wise as well, like giving it to researchers and everything for free eventually. But then with an agreement that, if you commercialize, we make some money there. We get a royalty there, whatever it is. And I think that will actually lead to drugs being developed even faster, and thus even more revenue raised even quicker. So that's great. And for humanity's sake, that's just terrific. My last question, this is, well, not my last question, but my last series of questions. This is for Kristy again. What what other indications are we going into, and like how can the similar kind of technology that we see with like Duchenne, like how could that be applied to other indications? I know I read like looking at like diabetes and things like that. So I'm curious, just kind of get an overview of all the different areas that we want to go into.
Yeah, so we have, as you can imagine, a tiered approach. So the DMD muscle program is our lead asset, and coming out of hopefully positive clinical data, you now have a way of making muscle satellite cells. So there's many other muscle diseases or injury that we could take the muscle program. So that in and of itself is a very robust pipeline. And then you can think of the pipeline going the other way of other cell types. And so our other publicly disclosed program is in brown adipose tissue. And so we have preclinical data showing normalization of insulin levels in diabetic mouse models. Obviously, metabolic disorders is a well-researched area and a hot drug development area. And so we think that there could be a place for this type of cell or organoid in a complementary approach. And then we're also looking at collaborations in the neuronal space or tendons, cartilage, as many protocols as Jonny can give me. We will find partners. And with each program, it feeds back more data into the model. So we can make our not intelligent more intelligent and more intelligent. I think we'll reach superhuman. I don't know. We're back to the philosophy. But the more programs we drive forward. And so some will take ourselves, some will partner. We're very flexible on that approach. We think that when you bring in experts into the whatever cell type you're doing, they can help also feed into the model.
So like, take for example, like paralysis, like somebody that gets in a car accident, they get paralyzed, they have nerve damage. Is that an area that we could theoretically go into one day with all this?
Yeah, I mean, there's tons of regenerative research in that area. So, I mean, you want to research a little bit more in there. But if it's a missing cell type that we can make that we think would make a difference, then absolutely.
Yeah, see, that's just very exciting. Like my whole cause of life, right, is to minimize human suffering and propel human prosperity so that humanity can have a better quality of life. That's like my mission statement for everything that I do. My why, like if you ever heard of the author Simon Sinek, I read that when I was like 15 or 16, and it stuck with me, and I found my why, and so that's everything I do is that. Speaking of that, I wrote a book which incorporates a lot of that. Micha, I know you said you wanted to talk about that, so I thought that's a good way to maybe end our discussion. So I give it to you.
Yeah. So first, I've only started reading it. Right, I received it yesterday. To be fair, Kristy brought it to me, and kind of throughout the podcast, it was clear first how profound and deep and kind of thoughtful your questions were. Also, how entrepreneurial you were thinking through this as a real businessman, and kind of I'll just say that I find this really, really, really moving and compelling. And yesterday, when we were sitting at Omnia, I was I was starting to read the kind of back of this to the people around the table. I had to stop because I was tearing up because it was just so inspiring to me. And so what I wanted to share is I forced Kristy to actually read this. I just think on a personal level, what you're doing is so beautiful, well beyond what you're doing for yourself and for people with Duchenne. I think kind of, people suffer and go through major challenges in life, even without suffering from DMD, right? Having, people go through a lot of things and over the last few years, I've been with people that didn't suffer from physical diseases, but had different ailments and other challenges in their lives. And, on a very, very personal, very sorry, at a very close to home level, just saying how motivating this is for people that are haven't heard the three letters DMD ever, and how transformational kind of what you've been doing, and kind of lighting a path for just really shining a path for and light on people going through their own challenges, and how personally motivating I find it, and how much it makes us work harder to help. We're not there yet, but to help kind of make this small, if a little bigger.
I really, really, really appreciate that, because that's something that I actually thought about today. I think about it quite often. Is I realize that Duchenne has made me more empathetic to people with all kinds of suffering. You know, I look at wars right now. I look at you know, people you know, in poverty, or you know, child abuse, like all these different things that are just horrific. And, you know, I truly want to go help there. That's why I asked about the other diseases. You know, I want to help all diseases, truly, not just Duchenne. I work on Duchenne because I have it, and that's what I got really good at knowing. But, you know, I just think that it's important that we use our suffering to help people overcome theirs, right? You know, I think suffering is something that every human relates to, whether they're rich or poor, whether they're, you know, smart or not so smart, whatever, wherever they are. You know, everybody has challenges in their life, some type of adversity. And so um so yeah, I appreciate you saying that, and realizing that, because that's really the cause of my life. It's really not Destroy Duchenne. Destroy Duchenne is the manifestation of what my cause is. My cause is to minimize human suffering, you know, to make people's lives better, and that manifests itself in a lot of different ways. So you know, it's truly a pleasure to be around all of you, to be a part of this team, because, you know, you guys are all highly intelligent, highly accomplished, all fascinating people, and you know, that's what I aspire to be as well. Um, so yeah, I appreciate that. Does anybody want to any last comments before we go and end this podcast?
It's been a pleasure.
Thank you.
Thank you for having us.
Thank you for having us, and thank you for doing everything you're doing. And looking forward to the next 3 hours that you host us.
Yeah. Thank you guys so much for listening to the Destroy Duchenne podcast.